Hello everyone, I'm Chaitra from Maastricht University and I'm going to present a method that we developed to identify characteristic features of metabolic states using genome-scale metabolic models. Metabolic dysfunctions are associated with several diseases such as obesity, type 2 diabetes, neurodegenerative diseases, and cancer. This makes systems-level understanding of cellular physiology very important for developing treatment and prevention strategies. In this work, our main interest was in studying the network-wide effect of different environmental perturbations. The primary objective of this study was to develop a method for comparing metabolic states. Our method identifies sets of reactions whose fluxes are affected by changes in the cellular environment. In this case, the uptake of certain metabolites belonging to branched-chain amino acids. As a biological use case, we applied our method on the model of human adipocyte and investigated the metabolic processes affected by blocking uptake of three branched-chain amino acids, namely leucine, isoleucine, and valine. We simulated two conditions labeled unconstrained and constrained, where the uptake of these metabolites was either unlimited or set to zero, respectively. Let's look at the method in more detail. To begin with, the flux spaces corresponding to both metabolic states were decomposed in three steps. First, the distribution of fluxes of all reactions were identified in both states. Next, the flux vectors explaining variation in the flux space were obtained through principal component analysis. Flux vectors are basically linear combination of reaction fluxes. Then, the flux vectors were transformed into biochemical space using basis rotation. These decomposed vectors were then compared to find those flux vectors that were biochemically distinct between the conditions. For this comparison, we used independent component analysis, or ICA. Finally, the reactions thus identified were reconstructed and visualized as a reaction network. Moving to the results, Figure 2 shows the flux distribution plots for two reactions involved in branched-chain amino acid metabolism. Compared to the unconstrained green curves, we observe that blocking branched-chain amino acid uptake has shifted the purple constrained curve to the left along the x-axis, indicating reduced flux. In order to explain the model-wide flux differences, the mean fluxes were compared between unconstrained and constrained simulations. This comparison is shown in Figure 3 and it indicates a strong similarity between the two simulations. There were few reactions which had either an increase or decrease in mean flux. Interestingly, many other reactions from branched-chain amino acid metabolism also showed reduced mean flux. However, this comparison did not reveal interconnections between pathways. So moving to Figure 4, we observed that following decomposition, 200 out of 4067 flux vectors were able to explain 75% variation in both flux spaces. Although the variance explained was very similar between the simulated conditions, there were still differences. These differences were then extracted using ICA and transformed into biochemical space. Figure 5 shows the reaction network containing the distinct processes after ICA. Every node is a reaction and every connection is a shared metabolite, either as reactant or product. These nodes are color-coded as per the simulated condition. The network is dominated by reactions active in one of the simulated conditions as indicated by the increased green and purple nodes compared to the orange ones. This shows that our method was indeed capable of extracting the perturbed features from the two flux spaces. A closer look at the biological processes involved revealed several interesting aspects. The branched-chain amino acid catabolic pathway was only active when these metabolites were available, shown by the green nodes here. Furthermore, connections between branched-chain amino acid metabolism, TCA cycle, and fatty acid metabolism were also found. C13 labeling studies have shown that the products of branched-chain amino acid breakdown entered TCA cycle and fatty acid metabolism in adipocytes. Our simulations also showed an overall change in metabolite uptake release profile and a close relationship between metabolism of branched-chain amino acids and other amino acids. Our preliminary findings support the idea that the method described here can be used as a hypothesis generating tool, which can potentially be verified experimentally in a lab. Finally, our method is scalable to other biological systems and therefore opens up opportunities for exploring and comparing other metabolic conditions. With this, I would like to thank you for listening.